day you want for final exam. So yes, it's correct that we don't choose when are we going to have an exam. So faculties cannot choose that. However, so university schedules an exam based on what students say, and then we have an option to change the date, but we have to go through literally administration hell. So current data that you guys have and that I still don't see. So my web track does not show December 12th, which I don't understand why, and that's why I'm getting in touch with scheduling office. You have December 12th. In syllabus, I put December 20th just because I wanted you guys to have extra time to study. That's why there is no class on December 12th, so you can sit at home and digest everything. So I don't mind. You can have exam, but I will change the date if majority of you wants. Like if most of people say they would rather prefer December 20, then I will start all process because it's not easy process. So in case that I have like 50-50, then we will stay with December 12. That I know that doesn't work for many people. There is the third exam on January 5th. And if I am approved, which again, I don't know if it is going to be possible, I would be able, I'm fine with having additional term for final exam. I'm okay with that. So I don't mind. That just means like I have to ask chair, is that allowed? Because I don't really know and I want to follow the rules. So for now, let's stay with December, that whatever is on bear track. And as you are voting, I will see how things are going. So if majority says December 20th, then we will change it to December 20th, but I have to go through all process. Now, I'm out of curiosity, how many of you here wants 20th over 12? Woo, many. You and, well, yeah, so there are many of you who actually cannot make it on December 20th. That's my fear. Yes, say you, sorry.
Oh, yes.
Hmm?
When I was looking at it, I was happy on doing it. Probably they also have exams. Yeah.
Yeah, I know that. Like, if I organize the exam on that, that's already disconnect. Yeah, okay. So yeah, you can. I can also examine 20 or for my other class, microbial physiology. So if there will be an exam on 20, it will definitely be in the morning because I have another exam at 2 p.m. So whatever there are, and it's 3 hours in there. So you won't need 3 hours. You won't need 3 hours, sure, but I give you an extra time. So again, think about that. Not really, not. You can be completely honest. And my job is to figure out, is there an option for additional term on 20 if majority of people say 12. So if not, there is the third exam on December 5th. Just sign up as soon as possible. Not December, sorry, January. My bad. So anyway, till now, so everything what we are talking today and on Thursday does not go on quiz 1. So lectures 1 to 6, quiz 1. I think I got a question on forum and probably just didn't click submit, but I will. I will just tell you now for the quizzes. The question was whether quizzes will be multiple choice and only you should study only from review sessions, and for midterm study from lectures. So I'm not able to tell you what I will ask you. That you get it. Right? That's why it's exam. But when I make my quizzes, I'm using the review session. So now you figure out what I want to say. When I make midterm, I will use my lectures. And for midterm, questions will be a few sentences to write. And because it will be an hour and a half, so you have more time for quiz, mostly I give multiple choice. And I can tell you that I have only multiple choice. So, there will be questions for like easier harder, but because I have to make some sort of selection. So just read it carefully, and you don't have to come back for quiz. So you finish it, so whoever is doing it in Academic Success Center can go home afterwards, because lecture that is scheduled for that day will be recorded and posted. That's a special lecture that I just wanted to give you as additional information, but you don't have to come back for it, okay? Now, oh, okay. For the name of God. So we talked about metabolic, yes. Yeah, so okay, I will just repeat it for recording. So the question is whether quiz is in this lecture hall. Yes, quiz will be here. So everything will be here. That's how they scheduled it. So the only, if final exam ends up to be on 20th, which I really don't think so, but I will do my best, then we will have to think about scheduling the room. And they told me that that's actually one of the problems, because they already scheduled rooms for everything. And if I come and say, okay, I want to do on 20, then they have to reschedule everything. That's why they don't really want to do it. But I'm willing to go through that. So if they approve, I will deal with that for my kids. Yes.
Do you have a practice problem?
So the practice problems are in the book. I can announce it. So the book, Brock's Biology of Microorganisms, at the end of each unit has practice questions. So if you guys want, I can just take them and post them in Word. Yes, okay, let me write that down. These are more, so these are more for like for you to practice, like to see how, like a literally like a summary. But again, I make questions, and I will post this in Word document under the review questions under each unit. That is not a pen. So I will write it later. Please remind me if I don't post. Yeah.
Last question. You mentioned some things that were like mentioned in lectures that would be on the quizzes, but they're not in the review section or like the review booklet. So
Then yeah. Okay. So I can tell you what will be on quiz. So, but I mean, you're asking me pretty much impossible thing. That would be breaking an ethical code. I cannot tell you what I will ask you, but if I maybe told you that something that it's important to know, but that might be important only for this term. I gave you review for quiz. I think that's already more than enough. So you guys, and I think that a review, in my personal opinion, review slides will make no sense if you don't read lecture. Review slides are there to help you to narrow down your knowledge and check up yourself. But if you really want to, I personally wouldn't be able to learn only from review sessions, because if you don't know background, then how will you, like, it's pure memorization, and then what's the point? Like, you have to read lectures. So I hope you understand that. And I cannot really tell you, if I tell you what will be on quiz, then why do we have class? Because I can give you all 8 and let's go home, which will be ridiculous. So again, trust yourself, it will be fine. So any more questions? Sorry. Okay, we are good. May I ask why you are all scared of quiz so much? Why? Yes, Yuan. You are not offensive, tell
I think that's because people look at emoji everything's you're supposed to study everything. So you're not supposed to like memorize.
you know, but what kind, so I'm not sure how to, like, how to get exam without memorization. So you guys are getting university degrees. So there must be some, I'm not going to ask like how many electrons are transported or those kind of stupid things that you can read everywhere. But how can you understand the basic of life if you don't know why is RNA here, why is RNA world? one theory. So you have to memorize that there are 70 S ribosomes. You have to memorize that the 16 SRRNA. So that's the level of memorization. Like, and there are logical things. Like, if I tell you that something is desulfovibrio, that means that it does desulfurization or sulfur removal. So that you have to, that's basically understanding. If something is anamoxidants, that means it has anamoxidant. So you have to memorize something. I'm trying to get it as easy as it can, and that's why I'm making that as a story, so you can. That's why we have that like first metabolic diversity, so we understand how life formed and which were metabolic pathways, and then we didn't talk too much about bacterial and specific species. But every single species that I give you will be literally something interesting that you can memorize so you can remember that. For example, Pyrococcus furiosus. I mean, it's a very unique name. It's furious, like fire furious. So that one is doing proton respiration. So those kind of things. So some level of memorization must happen. So that's how you select good from bad from the best students. So I'm trying to do my best to help all of you to memorize it and to understand. So I'm more for understanding, but some things have to be memorized. Is that okay? So we will talk a little bit now when we finish with metabolic diversity. So we know that there are different metabolisms that evolve in different times. But there are different types of diversities that we are covering. So metabolic was one of them. And that was important to understand how life formed. In terms of functional diversity, so here you will have organisms that are maybe phylogenetically, so they're belonging to the same group. They're all bacteria or they're all archaea, but they are functionally very diverse. One of them is doing one type of metabolism, the other one is doing the other type of metabolism, and all of them perform different functions in environment, and that influences their diversity. So today we will cover diversity of phototrophic bacteria. If we have time, I will finish again a little earlier. I always want to give you an extra time because I will, if I don't finish, then I will re-record myself. And then we will cover basically diversity in OVS cycle. So we'll talk about mostly bacteria here, because there is all group when I will talk about diversity of archaea. And that one is not easy. That's why I don't want to torture you too much. So in terms of microbial diversity, that can be observed, as I said, phylogenetically. And phylogenetic diversity, you will see when Nora explains to you how to build phylogenetic trees. So that's where 16S rRNA comes into the game. So that's where you distinguish bacteria from archaea, different types of bacteria. But you have functional diversity. So that's a different thing. So phylogenetic diversity means diversity in evolutionary lineage. So among bacteria. So what's diversity among bacteria? That's phylogenetic. We talk about one phylogenetic group. So that's based on 16S rRNA. If we talk about form and function, then we talk about physiology and ecology. So they perform certain metabolic pathways, metabolic function, and that's the role that they have in the environment. So whatever they create helps other bacteria to grow, so they form like a diverse community. That's why functional diversity is quite different than phylogenetic. There are two components, metabolic and ecological. For example, why we group microorganisms functionally? Because if they share common genes, that usually means they share metabolic characteristics. So when they share same metabolic characteristics, regardless of whether they are bacteria or archaea, they play similar ecological role. So in that case, it doesn't matter do they belong to bacteria or archaea. They have same ecological role because they have same metabolism. Now, for example, the organisms that share those ecological roles can be in the same evolutionary lineage. So, for example, there are phototrophic bacteria, and there are no archaea that do photosynthesis. So, in that case, so we say that functional diversity is very similar to phylogenetical because you have only phototrophic, so only one lineage phototrophic bacteria. Or it can be in different evolutionary lineages. For example, you have hyperthermophilic organisms, so organisms that grow at high temperature. There are bacteria and archaea. So evolutionary, they are very diverse. They both diverge from Luca and they evolved separated. But then they had like a literally adapted to perform the same function because they adapted their metabolism. And there are various reasons why there is that difference between phylogenetic and functional diversity. So some of them are listed here. So why would something be different phylogenetically and functional? Why that not say? So it can happen that gene is lost during evolution. So maybe gene was present in common ancestor for like Luca, several lines. So some lineages kept it, but other evolved to lose it. And then they have that discrepancy. So they're in the same evolutionary line. So they're all bacteria, but some of them have functional traits, the other ones don't, depending on where they live. Pretty much depends on what was available for them to metabolize, to generate energy. The other reason is convergent evolution. When trait involves, when trait, one characteristic evolves independently in two or more lineages, or horizontal gene transfer. So horizontal gene transfer is something that was very extensive early, but as the barrier was formed between bacteria and archaea, horizontal gene transfer was lower and slower between different lineages. So in beginning, that was very extensive. So basically, that would mean that horizontal gene transfer was, horizontal gene transfer was possible between bacteria and archaea early, but as they evolved more, that AGQ was not. In terms of phototrophic bacteria, again, there are only bacteria. When we talk about hyperthermophilic organisms, we'll talk about archaea a lot, especially when we talk about diversity of archaea. So many of these things will be only repetition of what you already learned in metabolic diversity. So phototrophs are using light and convert it to chemical energy, and they evolved very early. We know that they were on the bottom of phylogenetic tree. And there are 5 phyla of bacterial phyla, and I will ask you to know 2 of them. And those are Chlorobia and Chloroflexi, because those are the most important. Protobacteria, so this is anoxygenic photosynthesis present in these 5 phyla. Chlorobia and Chloroflexi are the most important. Protobacteria are the most diverse phyla, so protobacteria are not only anoxygenic phototrophs, there are many others. That's why I'm not going to bother memorizing it. So, but these 2 are very important. Oxygenic photosynthesis exists only in 1. So that's cyanobacteria. So you can, and I purposely put these 5 in alphabetic order, so that because that's how I memorized it. But again, Chlorobia and Chloroflexus would definitely be the one that I will pay the most attention. And of course, cyanobacteria. Now, some common features in phototrophic bacteria are chlorophyll-like pigments, many accessory pigments, and light harvesting complex. So that's common for all of them. Simple repetitions from what we learned. And in terms of that one phyla, oxygenic phototrophs, cyanobacteria, this is something that you probably mostly heard about, but there will be a few organisms that I will tell you about, and I will tell you why I would like you to learn about that. So first of all, they were they're first organisms that generated oxygen on Earth. So they actually generated a basis for eukaryotes to develop. So they were those that converted Earth to oxygenic environment. And they're very diverse in morphology. So diversity of cyanobacteria is really impressive. Like pretty much you can find whatever you want in cyanobacteria. And I will show you 5 different morphological groups. They have different functions, and that's why they are different. They're shown, they're presented as a different organisms. So mostly they can be unicellular or filamentous. So that would be broad division. If they're unicellular, they can either divide by binary fission or multiple fission, but they are unicellular. Unicellular one are pleurocapsales and chlorofocales. So chlorofocales are those that divide single, and pleurocapsales are those that have multiple fissions, and this is how their division looks. So that's how you will recognize them in the microscope, on the microscope. Filamentous are my favorite. They are really, really, again, even though they're diverse, they're all filamentous, they just have different characteristics, and based on that, they perform different functions. So, but they're all filamentous. So oscillatoria, you probably heard about this when in one of our first lectures, where we talk about oscillatoria linetica. That is an organism that does it's oxygenic phototroph, but it can also adapt to do anoxygenic photosynthesis. So that's how oscillatoria looks. The one that is more famous is Nostocales, or Nostoc. Nostoc is a genus. So this one has known cellular difference. So there are different types of cell over there. It's filamentous and has different types of cell that make that filamentous. The only characteristic of oscillatoria is that it doesn't have heterocyst. And heterocyst is a type of cell that I will show in a few minutes. Now, the last but not least is Stigonema or Stigonematales, which has branched cells, also filamentous. And that's the last one. So I first hear something. No, okay. Can somebody please close the door? Because I think I hear from other class, you might think I hear people. So anyway, so are cyanobacteria group coherent? So are all of that, pretty much what that means, do they also have, do all members share common features in cyanobacteria? Well, no. We just saw there are 5 groups. So they all perform different functions. They all have same metabolism. They're all oxygenic phototrophs. They all belong to the same evolutionary lineage. So phylogenetically, they're all cyanobacteria. But functionally, they're quite different. There are 5 different groups. So they perform completely different functions, and some of them are very useful for us. So the only is, yes.
When you say coherent group, do you mean like they're sharing common features because of common ancestry, or because they're evolving with common features convergent?
The second one, they evolve common feature convergence. They all share common ancestor. Yes. So thanks for clarification. Yes. So if you want to think about which one is coherent, so one among 5 groups is coherent. Like the one Pleurocapsales is the only one that has multiple fissions. So members of that group are coherent. But when you look at that group comparing to the other 4, they're not coherent, because so that means that this multiple fission feature arose only once in evolution. Didn't happen again. Okay. Then then you think about those filamentous bacteria. So 3 groups. All 3 are different, but they're all filamentous. They all share common ancestor. So and they all have single origin of differentiation. So they are all filamentous. The only group that has this branching is Stigonematales. That means that feature arose only once in evolution, the same like multiple fissions in Pleurocapsales. And these are some key genera that I am pretty much not. I don't think I will ever ask you this for exam. maybe for quiz, because this Synechococcus is amazing organism, and there are great review papers on Synechococcus. Many things are done in terms of biotechnology with Synechococcus. And later on, we will mention Trichodesmium and oscillatoria, so it's going to be easy to memorize. Some major characteristics of cyanobacteria you already know. So this is simple repetition of what was done in previous. So they have photosystems 2 and 1. That's pretty much evolutionary advance comparing to anoxygenic organisms. Carbon dioxide fixed via Calvin cycle, the one that is most energetically expensive, evolutionary youngest, but the most spread. Some of them can be photoheterotrophs. So that means that they can even use sugars when they don't have light. So I'm not asking you which one. So I'm pretty much focusing on this Limnetica because that one can use sulfide. So it's the example of oxygenic phototroph that can do anoxygenic photosynthesis. Some of them can fix nitrogen, which is great. Then we'll talk about their special cells, usually those filamentous ones. Some of them can synthesize vitamins, and I think about this from biotechnological perspective, how can you exploit that feature? Again, some of other characteristics, pretty much repetition, thylakoids. So they're the only one that have thylakoids, like internal membranes. Anoxygenic organisms do not. And the cyanobacteria are the closest to the plant. So there are some, those are special membranes that literally concentrate pigments and increase light harvesting activity. That's all what I want you to know from that. And now they have some specific structures that we never talked about, and I pulled up some interesting research on specific structures of cyanobacteria that I found interesting. If somebody has another idea, please let me know. So they have extracellular structure that is helping them to move. And that filament is also suitable for gliding motility. So we will talk about gas vesicles, envelopes, different types of cells. So this we will cover basics of everything. Again, if somebody wants to know more, I can definitely give you more information. So what is filament used for? So filament is helping cells to move in the environment. So basically they slide, they glide through the environment, and what needs to be done here, you need to have a contact with solid surface. So they can, in order to perform their function, they have to somehow move. So they have to very often move toward light. So they need light to perform photosynthesis. So they can either glide, like move slowly toward surface, or they can perform rotation. So they can do both. Phototaxis is what, so phototaxis means moving toward the light. So in order to perform their function, which was photosynthesis, so, generation of oxygen, they had to move toward light. So they have developed those gliding motility structures and rotation, so they can get to the place where there is the most extensive light. Now, the fun fact, this is from a paper that I cited below, so you can find more. So what they found, this is pretty much old paper from 1998. So this is, they call this pore complex actually secretion organelle. So cyanobacteria has something that reminds on organelle. So that tells you how close they are to the evolution of like eukaryotic organisms. So this pore structure resembles organelle. So that's what they use. So that's like a molecular motor that helps them to glide. So this motor is on the surface, and this is just called like a microscopic image, electronic microscopic image of that pore complex. So it's basically in cytosol. So it has like a structure that is pseudo-organelle in cytosol, and it spans through all membrane, and through that pore they secrete polysaccharides that help them to bind to the surface. So it does look like an organelle. It's a form structure, but it's not, that's why I call it pseudo, because it's not real organelle in prokaryotes. So when they observe, so what those people from the paper who published this paper observed is that this pore complex is actually a site of secretion. So I think they tracked it in real time. So they had labeling stuff, so they could track how the bacteria is moving as it's secreting that polysaccharide through that pore complex. So it pretty much looks like a real organ. Any questions? So that's a gliding motility pore complex found in cyanobacteria. In terms of gas vesicles, so this is something that is also very important for cyanobacteria, and everything is developed to help them to reach the area of the best environment where they can do photosynthesis. So these gas vesicles, when they produce them, that helps them to float in water, and then they can go to the surface where they can reach more light. So again, they are only they're only for positioning cells in water. So again, everything with a purpose to get where there is the most of light, because they need light to generate energy. So that's all purpose of these gas vesicles, nothing else. Again, fun fact, these gas vesicles, this feature, evolved in cyanobacteria to help them find a food. We use this feature to develop biomedical engineering. So, because what those vesicles do, they can carry oxygen. That's the point. So they carry gas. So what people who do biomedical research done, they did some engineering of proteins that are attached to those vesicles, and they made them efficient oxygen carriers for biomedical treatments. So again, just a fun fact. That's how I would like you to memorize it. That's why I'm giving you a fun fact. So Anabena, one of photosynthetic bacteria, one of those 5 genera, has those gas vesicles that increase oxygen that have oxygen carrying capacity. So what people thought it would be great, and they actually did it. Please don't memorize these proteins, but they just engineered proteins that are on the top of vesicles. So this pretty much looks like a mini cell in cell, right? So they just made those vesicles and they decorate them with proteins. So when they engineered those proteins, they actually increased the oxygen carrying capacity. Again, just a fun fact, don't memorize this. This is from paper. I'm just giving you an example how feature that evolved with one purpose was exploited by humans to make a better oxygen carriers, because then you can have better oxygen transport in biomedical research, because when you work with cell culture, you need efficient oxygen transport. Again, just for the fun fact, Anabena is the one whose gas vesicles were used in transport, in engineering. Questions? Now, mucilaginous envelopes. I don't pronounce this right, but basically that means bound filaments. So one filament plus one more filaments at giving that envelope, which is another structure that these organisms develop, and it helps. So this is how it looks. It actually, so they secrete more exopolysaccharides, and they make like a gelatinous structure, and that groups cells together. So when you look them under microscope, they don't exist like a single cell. They exist like a multiple formations of, like a formation of multiple cells. So that's because when they are growing, they secrete polysaccharide. So pretty much same like filament, just this is more filaments. So more and more filaments they make, they have this structure. And the purpose of this is protection. So cells secrete exopolysaccharides so it can protect them from losing of water. sometimes that's the way how they protect from from toxins, from predators. Biofilms do this. So cells are organized in these structures called biofilms. And you probably heard about that, like there are biofilms that form on lungs. I think it's Pseudomonas, and then that causes a big problem because then antibiotic cannot reach them. So the reason why they like to form those biofilms is to protect them. So the multiple cells that make this gelatinous substrate that helps them to prevent antibiotic from coming in. Just an example from a paper. This is emulsion. Basically, this is another type of exopolysaccharide that bacteria make, but they don't make it to protect themselves from predator. They make this polysaccharide as a population, and then that helps cleaning a space so they can get more light. So that's another interesting point of evolution. They have another types of specialized cells that they use for dispersion called hormogonia. So this is the picture of hormogonia. Basically, there is that short motile filament, and the point is that it can. So this hormogonia is pretty much easily separated from cells. So when there is a time of stress, where cells are stressed out, hormogonia will detach and let mother cells to die, but then it will migrate forward. So that's how population is maintained. So simple to disperse from the stress. These akinetes are used for protection, and you will notice that most of them are used either for protection, either for predation, or for adjusting themselves toward source of food. And akinetes are very similar to hormogonias. They're basically just a thicker cell walls. So the cells form cells. So if you heard of, yeah. Stressing?
Like when the cell stress, how does it impact?
Okay, so do you mean hormogonia? Hormogonia? Yeah. Okay, so hormogonia is like, so this is when you have filaments of cells, they're expanding. So when that's pretty much like correlation in bacteria, if you guys heard. So what that means is when there is some sort of stress, let's say there is a toxin or something, that signal initiates something that is very similar to cell division, just it's not same mechanism of regulation, because that cell, so basically that cell literally divides horizontally. So filament splits it too. So it's that cell that is separated from hormogonia is different than, so it's like a cell division, but forming a different type of cell. And that's caused by stress, because if there is no stress, cell would regularly divide into well. In this case, because of the stress, that hormogonia separates from the mother cell, and then it migrates. How it migrates? Because they usually they have those pores where they secrete like exopolysaccharide, and that can help them to secrete it, attach. So like a jumping. Some of them can also rock it. But when that happens once they are under stress. Does that answer your question? Okay. Any more questions? Can you imagine that thing? There are very nice, so many of these things are very nicely shown on YouTube. So if you Google on YouTube, it's like people now have electronic microscopes where you can actually record what bacteria are doing. So it's quite interesting. That's how I memorize it. When I see it, I understand it much better. More questions? Okay. And akinetes are similar like hormogonia, just they have thicker cell wall. That's why we call them thick, like cells with thicker wall, but they're similar principle. And usually they are smaller in size. They're like shorter. Okay. And last but not least, for cyanobacteria are those two structures that are particularly important for us, especially when we talk about heterocysts. Those are storage structures. So cyanophycin and heterocysts. Heterocysts is something that you probably heard, but you were not aware that that originates from cyanobacteria. So what's the purpose of these two structures? Storing a nitrogen. So remember how I said that these organisms are very diverse in terms of like they are phototrophic, but many of them can fix nitrogen, like atmospheric nitrogen. So atmospheric nitrogen fixation occurs in these specialized cells, heterocysts. And some of them have this cyanophycin, which is like another way how they accumulate nitrogen. So literally they make, so cyanophycin is accumulation of amino acids, aspartate and arginine. So that's not from nitrogen fixing. But heterocysts are storing nitrogen that originates from nitrogen fixing. So this cyanophycin is something that basically those accumulations of amino acid help for cell to, when cell is starving for nitrogen, it just takes out those amino acids as a sort of source of nitrogen because there is no external nitrogen source. So same way, if you heard about ketone bodies in our organisms, so when we are on restricted diet, we generate a bunch of ketone bodies because that's how our organism gets energy. So we are storing it. The same is what they do just with cyanophycin. So sometimes we can have carbon storage, nitrogen storage, but when we try to lose weight, we try to exhaust our internal carbon and nitrogen supplies because we don't get anything from outside. So the same they do with nitrogen source. Way more fun are heterocysts. Those are specialized cells that are meant to do nitrogen fixation. If you think about that, have you ever heard for leguminous plants? You don't know what is leguminous plant? You heard. So those plants have symbiotic relationship with those bacteria, with cyanobacteria that fix nitrogen for them. So basically, plant provides them protection. We will see against what, and bacteria fix nitrogen that they use to plants. So what is so specific about heterocysts? So they're present in nostocs, in nostocales, and they're usually different cells that are present at the end of filament. So they develop from regular cell. Regular cell is vegetative cell, but they're different because only in those cells nitrogen fixation occurs. Why only in those cells? As we will see next slide, nitrogen fixation, so people used to thought that nitrogen fixation was one of the oldest metabolic pathways that evolved. Nowadays, people say it's actually hydrogen metabolism. So nitrogen fixation, the reason why people thought that nitrogen fixation is one of the first is because it's extremely oxygen sensitive, which means in order to do fixation of atmospheric nitrogen, you need to have zero oxygen. So if it happens in cells that are doing photosynthesis, which cyanobacteria are doing, so they're doing photosynthesis and they make oxygen. So what you have to have specialized cells that will provide certain protection of oxygen, which means those cells have to do modified photosynthesis. So that's why those cells evolve some thick cell wall that will prevent oxygen from entrance. And again, the enzyme that does this fixation of nitrogen is called nitrogenase or denitrogenase. I won't ask you these details. So, but this enzyme is very oxygen sensitive. So nitrogenase cannot be active in regular cells. It can only be active where you have no oxygen. So how those bacteria evolved anaerobic and aerobic processes in the same organism, and why do they do that? So it's very possible because they developed regulatory mechanisms that that basically ensure that you separate activity of nitrogen fixing and photosynthesis. How? So if you think about two photosystems that are doing that are in cyanobacteria. So oxygen is made only on one photosystem, photosystem 2. So in heterocysts, where we don't want oxygen in heterocysts, cells don't have photosystem 2. They're very similar to, so only photosystem, you have only photosystem 1, which is similar to green sulfur bacteria, which we'll cover later. So they can, because they don't have photosystem, that means they cannot generate energy to fix CO2. So pretty much the function of heterocysts is only to fix nitrogen. When they develop symbiosis with plants are and this is something that I'm just giving you as the information. I'm not going to ask you that, but it's symbiosis. So plants provide additional protection from oxygen, and they fix nitrogen for plants. So that's how symbiosis functions. Some of exceptions in nitrogen fixation is that, this is not basically, so when we talked about heterocysts, so that's adaptation, like that works for those that have filaments, so multiple cells. If you have cyanobacteria that have only one cell, unicellular, they also do nitrogen fixation, but they cannot separate it spatially because they have only one cell, so everything must happen there. So what they did, they evolved how to fix nitrogen only during night, when there is no light, there is no photosynthesis, there is no oxygen produced. So that's called temporal separation. So spatial means you separate in space. Temporal means they do it different times. So different time they fix nitrogen, different time they do photosynthesis, but everything happens in the same cell. And this one is quite interesting. I don't know many things about this, but this trichodesmus apparently reduces the amount of photosynthesis when there is nitrogen fixation. I don't quite understand how is that happening. There is probably molecular mechanism. I didn't find any interesting research to share, and it would be too much. So knowing about heterocysts will be really fine. Again, is it early in evolution? So people think before this new paper that I will present at the end of Unit 3, hydrogen metabolism, people thought that nitrogenase was one of the first enzymes that arose at the end of Archean in that ocean before oxidation of atmosphere via oxygenic photosynthesis, which were cyanobacteria. But as evolution was progressing, so basically evolution of cyanobacteria was progressing, molecular oxygen developed, species developed protective mechanisms to keep nitrogen fixation, but still do it with the oxygen present. That's how that's how it was shown earlier. And phylogenetic trees that are made based on denitrogenase gene, so this enzyme that does fixing of nitrogen, shows that there is very early branching, which means that it did develop early, but probably wasn't the first one, which we will see in the end of this unit. So nitrogenase was first, and then heterocysts evolved. So basically, eventually they lost photosystem 2, but nitrogenase developed first. Why they're significant? Well, because did you ever hear about algal blooms? So that's based on cyanobacterial activities. And so you will see that in waters, and there are many metabolic products from cyanobacterial. They can produce, so that algal bloom is actually not very nice for cells, for us, because it can be very toxic. It can kill animals. They're very often responsible for odors in specific areas. And one thing I forgot to say, cyanobacteria are a great source of third-generation biofuels because they have so much lipids. So that's, again, in the time teaching applied microbiology. Any questions so far? Clear? Okay. And the way how I divided this is basically purple and green bacteria. So we talked about purple and green sulfur, non-sulfur bacteria when we talked about summary of metabolic diversity, which is Vinogradsky column. So that order in Vinogradsky column you have to know. That's easy on the review. So who goes where? Now, now I will talk about purple bacteria that are sulfur versus non-sulfur, and then we will end up with green bacteria that are sulfur versus non-sulfur. They are all phototropic. So sulfur bacteria, as we know, are using hydrogen sulfide to run photosynthesis. And to correct myself, one of my slides says S2 as a product of sulfur oxidation. That's incorrect. It should be S0. So elemental sulfur. So when they oxidize sulfur, they produce elemental sulfur. And it's usually present in anoxic zones, so where there is no oxygen and there is a lot of hydrogen sulfide. And so these guys, purple sulfur bacteria, accumulate sulfur granules inside the cell. That's different than green bacteria. They do it outside. So that's important difference, but we'll come to that point later. I will mention it again. And non-sulfur bacteria are very similar. They still need light as a source of energy, but they do so. Non-sulfur bacteria do not fix carbon dioxide. They use organic carbon. And there are two families that they have. And again, since I really don't like to make you memorize many things, I would like you to know to recognize when you see picture, and I do want you to know where are sulfur granules shown, but I don't really like memorizing Latin names. So if you can avoid it, I will do my best. So chromatice and ectotheriorodospirice, don't bother, you will never know how to spell that. I also don't know. So, and you can always Google it. The way how you will memorize is that thio, T-H-I, that stands for sulfur. So that usually thio means sulfur, but I will never ever ask you to write this. I don't know how to spell it, so I have no intention to make you memorize these things. So, but you can memorize that there are two groups of purple sulfur bacteria, and it's important that these species, species in these two groups of bacteria are distinguished by location of sulfur granules and photosynthetic membranes. So again, you don't really have to know what are they called, but where is the difference between them? So chroma organisms, chromatice, they have granules, sulfur granules inside of periplasmic space. The other one, thio, has outside of them. And this one is exception because usually green sulfur bacteria have that out. What is important to know is the difference in photosynthetic membranes because one has vesicular, the other one has lamellar. But I might ask you to recognize them from a picture, which you will easily, maybe not because of colors. I will figure out, but I really want to reduce the amount of Latin names that I will ask. So for now, just know that there are two groups of bacteria, sulfur bacteria, that are different based on the way how they organize their internal membranes. Is that okay? Non-sulfur bacteria, again, they use organic carbon, so they also need light as energy, but they use organic carbon. And the energy conservation can be done in two possible ways: fermentation, anaerobic respiration. So, and for that, they need organic carbon. Either they ferment organic carbon or they use organic carbon to respire something else. And we talk about that. Key genera here is Rhodobacter. This one is very nice. And we already talked about this one when we talk about losing one of traits in nature. We talk about losing pigments in first lecture. We had two examples. So Rhodobacter is one of them. That concept might come up. So this one belongs to the purple non-sulfur bacteria. Any questions before we move to the last one, green bacteria? So for the purple bacteria, sulfur, non-sulfur. Purple sulfur bacteria, granules, usually inside of cell, two families, difference in two families, membranes. One has vesicles, the other one has lamellas. The one with tile has lamellas, and that's it. And in terms of non-sulfur, Rhodobacter, organic carbon, anaerobic respiration and fermentation. Now, green bacteria, again, sulfur and non-sulfur. For green sulfur bacteria, they also use, like purple bacteria, hydrogen sulfide. They also need light, and they also make as elemental sulfur, but they deposit granules outside of the cell, unlike purple, which do inside the cell. Interesting thing about green sulfur bacteria are specific types of bacterial chlorophyll that are organized in chlorosomes. Chlorosomes are those sensitive structures that evolved so they can literally catch all sorts of light present. And so let's go to purple sulfur bacteria, and then we will talk. So then we'll talk about non-sulfur. So remember at the beginning when I mentioned two phyla, Chlorobi and Chloroplexus. So this is where they come into the game. So Chlorobi belongs to green sulfur bacteria. You will memorize it based on chlorosomes. So it has chlorosomes. So basically pigments are organized in chlorosomes. Chlorosomes are accumulations of pigments. The most simply said, they're bound by tiny membrane. They are attached all the way to the periplasmic space, to the membrane. So you see that the arrow shows where chlorosome is. So they're very sensitive. That's why they are very sensitive, because they evolved in areas where they didn't have a lot of light. So they have developed very sensitive structures, so they can collect everything. That's why they have that chlorosomes. Non-sulfur bacteria are chloroplastic film, and they cannot be easily metabolically characterized because you cannot cultivate them. So that's why there are no many things that you can say about them. I mean, probably people who are doing ecology know many things. They are filamentous. That's the only thing that I can say. And they all belong to the phylum Chloroflexi. They can use organic carbon source or hydrogen. So there are two genera, which, again, since I'm not really fan of Latin names, I will not bother about that. But maybe like when I tell you some examples, you might memorize it. So these are two interesting, so especially thermomicrobium. That one is very interesting because it's the only one that has different structure of lipids. So let's say that this one is interesting. So, and it's not either ether or ester linkage. So this one is neither bacteria, it doesn't have neither bacterial or archaeal characteristics, and it belongs to green non-sulfur bacteria. I'm not sure how well it's cultivated, because I believe it's not that cultivation was very hard, because they did not really develop any research that I know about this organism. And cell wall is very protein structure. So that's, to me, this reminds more on bacteria, but because that lipid, the specific lipid structure is kind of unique. And that would be all for this one. So you again have 15 minutes for questions. I will now stop sharing and stop recording so you guys can ask